All right, hello everyone. We're going to continue discussing the chapter 27 on protists in this lecture, and we will be finishing up this chapter in today's lecture. So we finished up talking about a few of the evolutionary mechanisms from which, eukaryotes, organisms with a nucleus, arose from. So we're going to continue talking specifically about things that make up our protists. Again, these simple eukaryotes. So in terms of support and protection, we see that there's a whole lot of variation among the different lineages of protists with respect to things that allow them to be supported and to be protected. So some protists have cell walls. Some have these hard external shells, almost glass-like. We'll see rigid structures that are inside of the cell membrane as well. Um, so this diversification of protists has been associated with the evolution of many structures for support and protection. So lots and lots of evolution has led to basically the diversity that we see. Depending on the environment in which they live, there's different structures for support and protection. So let's look at some of those examples in different types of protists. So you can see here this diatom. Notice the shape of the diatom has this sort of glass-like shell and a box and lid arrangement almost, protecting this organism. Okay, so this is a type of cell wall, very glass-like, made of silicon dioxide. Dinoflagellates, these ones that can produce some problems, cellulose plates make up the outside of the dinoflagellate, again for strong protection. Okay. You can also see chalky chambered shells made up of calcium carbonate. Okay, so in this case, similar to a shellfish in the foraminiferan. Okay, so foramina meaning holes. You can see there's holes in there. The euglenids got this pellicle composed of protein strips. Okay, so here we see that with this, this pellicle, lots of strips that are located actually underneath of the cell membrane. All right, and we'll see different diversity in this as well and other types of internal and external support. These are just some main examples that your book goes over in this figure. Okay. So another evolutionary characteristic that has, happened over time with eukaryotes is the evolution of multicellularity. All right, so when we talk about organisms with more than one cell, like yourself, you're made up of many trillions of cells that all have different functions. It's multicellularity. So you have to think about what would make this, a really important adaptation. As we got more complex through evolutionary lineages, we saw that multicellularity started to dominate. Okay, you can now take on more advanced roles, more advanced mechanisms for obtaining nutrients, carbon dioxide, oxygen, all of those things. So selection pressures really led to this multicellularity. So it's hypothesized that colonial organisms, so meaning that you had your single-celled organisms that lived all together, started to take on different roles. And as they started to become so differentiated in their different roles, the cells stopped doing the things that they were doing before. So what happened was the cells became specialized for different functions. So not all the cells express the same genes. So the DNA in some cells in the colony only made the proteins that, were needed for one aspect of life, and some made other proteins that were necessary for other aspects of life. Okay, so this idea of multicellularity arose from tiny single-celled organisms clumping together and saying, okay, we can do this as a group now, and kind of share the load. And that's how multicellular organisms arose, right? This multicellularity arose independently in a wide array of eukaryotic lineages. So what that means is that in the green plants and the fungi, the animals, slime molds, and the red algae, it all kind of evolved at different times, depending on the selection pressure of the environment. So how do protists obtain food? So they have three ways of feeding, okay? So they either ingest packets of food, they absorb molecules directly from the environment, or they do photosynthesis, okay? So ingestive feeding, known as phagocytosis, basically you have your cell and it's going to, you can imagine, it's hard because you can't see me, but if you take your hand, you put it in a plastic bag and you take that plastic bag and you engulf it around something, that's phagocytosis. Okay, it's almost like a Pac-Man sort of thing. Absorptive feeding is eating live or dead animals where nutrients are taken up across the plasma membrane directly from the environment. This can involve decomposers, feeding on dead organic matter, or detritivores. Okay, many of these absorptive feeders live inside of other organisms. Makes sense. They're going to literally leach nutrients off of the digestive tract of another organism. This is called a parasite. Photosynthetic is plants. You can think about plants being photosynthetic, obviously taking sunlight via photosynthesis and turning it into glucose. Okay, so we have single-celled organisms that do photosynthesis, and we also have giant seaweeds like kelp that do it. Carbon dioxide obviously is going to be your major source of carbon from the atmosphere. Um, there's many feeding mechanisms that we've seen with this. And when we get into their movement here in a second, we'll talk about pseudopodia and cilia around the mouth and the predatory ways in which these things move. So here's an example of the pseudopodia, which is like the feet of the protist. Like I said, we'll talk about that in a second. You can see that the pseudopodia, this protist is literally wrapping itself around the prey species here and engulfing it. Okay, here's another example. The cilia around the mouth. Here's some food items, and this protist here has a gullet where it's going to take in these food items, and the cilia take the food items in, and ultimately they'll be digested in the gullet. So as the food items go down, they enact that cilia and they bring it in. So just two different ways in which protists can ingest food. And I have a couple of videos that I will post to Blackboard to give you some more idea of these things. Okay, so how do protists move? Okay, so why would protists need to move? They're going to need to actively find food, find light if they're photosynthesizing. All right. Amoeboid motion is a sliding movement produced by extensions of the cell called the pseudopodia, which we saw on the last slide. And cytoplasm is essentially going to stream into those pseudopodia. Okay. Pseudopodia, that term means like pseudo means fake, and podia means feet. So I also teach anatomy and physiology, and we see this pod prefix a lot of times have to do with feet or things shaped like the feet. Okay, so this amoeboid motion of sliding and pushing the pseudopods requires ATP or energy, very similar to muscle movement in animals. Okay, and key immune system cells in humans, white blood cells, use this same amoeboid motion. Again, I will post a video to Blackboard that will show you the amoeboid motions, so make sure you check that out when you get a chance. Okay. So here's an example of amoeboid motion via pseudopodia. Okay, you can see the movement here, the arrows going directly that way. And again, it kind of like is almost like a crawling motion. Like I said, I'll show you a video of that in a separate link, but very interesting, very cool the way these things move. Kind of nasty looking the way that they do move. So in addition to pseudopodia and that form of motion, the amoeboid motion, we also see cilia and flagella, which many cells use cilia and flagella. So flagella and cilia have some identical structures depending on the type of organism we're looking at. In all protists, they're going to be pretty much the same, but when we look at bacteria as a separate domain here, bacteria actually have very different structure to their flagella, but flagella and cilia have identical structures in protists. Flagella, though, are long tails that come off of the back, generally, of a protist, and also like a sperm cell has a flagella. It's an example of a human cell that has that. So you can have one long flagella, or you can have two, and we talked about the monoconta and the difference there earlier in the last lecture. We'll get to that a little bit later. Cilia are short and numerous. They're like little hairs that go around the outside of a cell. And what's interesting is depending on the environmental and selection pressures, even closely related protists can use radically different forms of locomotion. So it's not always the same, even in very related protists. Okay, so here's just again 2 examples, and I will upload some videos of these as well. Swimming via flagella. So here's the just the 2 tails in this case moving and allowing the protist to swim, and here's the movement going across the cilia, the tiny hairs. Okay, so reproduction, and these themes that we're going to see here with protists are going to come back again in the next couple of weeks when we do plants and animals as well. So you want to kind of pay attention here to these different life cycles because, like I said, they will come back again. There's two types of basic reproduction, and protists do both essentially. Asexual reproduction is making clones, exact copies, so based on mitosis, cell division in eukaryotes. In bacteria, it's called fission, but essentially it results in two daughter cells from one cell that are all identical. So asexual reproduction does not result in any differences at all in the DNA. Sexual reproduction, however, is based on producing these egg and sperm cells. So reducing chromosome number via meiosis, and then having those two egg and sperm come together. So in the process of meiosis, which you should have learned in Bio 1, you end up with a shuffling of genes through crossing over and independent assortment. So what do we get when we get this shuffling, and then we get the random meeting of egg and sperm as we get daughter cells that are genetically different from their parents and from each other. So there's certainly, advantages and disadvantages to each. Major advantage for sexual reproduction is you get diversity. So you see very different types of organisms, different susceptibilities to disease, and that's very important for evolution to take place. Asexual reproduction, you don't get any of that. So if, a disease comes through that wipes out the population, nobody's immune because you all have the same DNA. So asexual reproduction is very dangerous. The trade-off is that it's very fast, and you don't have to find a mate. There's very little energy involved. So some organisms have adapted to each of these situations, or some rely on both, which we'll see, like the protists. It's like I said, meiosis, very adaptive because of the genetically viable offspring. You have differences, and those differences allow for evolution. The genotypes in the DNA in general of many parasites and pathogens evolve very quickly. So natural selection is going to favor individuals with new genotypes that may be able to resist these parasites and pathogens. So say you're out spraying your lawn with weed killer, and you're spraying it and it's working, and all of a sudden it's not working anymore and your weeds are becoming resistant to that weed killer. That's because as you were spraying, it worked for a while, but maybe there was one weed that had a mutation that allowed it to be resistant to that Roundup or that weed killer. And suddenly that was the only one left, and it kept reproducing, and it's resistant, and that's the only one that's going to be part of the environment now. So you need to change your weed killer until the new population becomes adapted. But that can take very quickly, or it can take long amounts of time. Just depends on the how much genetic variability there is. Okay. I mentioned this earlier in the previous slide, but many biologists view sexual reproduction as an adaptation to fight disease, because if one disease comes through that kills the whole population and you all have the same DNA, your species is dead. But if there's some that are resistant, the species will continue. Okay, so let's look at some of the life cycles here that protists specifically go through, but we're going to see these life cycles in plants and animals later on as well. So obviously a life cycle is a sequence of events that occur as individuals grow, mature, and ultimately reproduce and repeat the process again. The evolution of meiosis, very important because it introduced a new event in the protist life cycle. It created a distinction between the haploid and diploid phases of protist life cycle. So again, depending on your Bio 1, what you remember, haploid means having half the number of chromosomes. We signify that with a little letter N. And diploid means having the full number of chromosomes. So all sex cells are little n, and they contain half the number of chromosomes. I always remember half the number of chromosomes. Diploid, double, or the full amount of chromosomes. So in humans, our regular body cells are all diploid. We're all diploid humans right now. We're 2n, 46 chromosomes. But our egg and sperm cells that we produce have half the number, and we say that they are haploid, 23. Okay, so we're going to see that very simple for a human life cycle. Okay, you have 2n, you produce egg and sperm cells that are n, they come together to make 2n, and the process continues. Okay, not always the same in plants and with protists. Okay, so in terms of bacteria and archaea, very simple. They're always haploid, okay? They're always N. They're always doing asexual reproduction. There's no diploid state to bacteria, okay? In protists, it's all over the place. Some are all haploids, some are haploid diploids, some go back and forth. It just depends on the species of the protist or the type of lineage. So to analyze a life cycle, start with fertilization, the fusion of two gametes to form a diploid zygote, and then trace what happens to the zygote. So you're not going to have to memorize these life cycles, but you are going to want to be able to interpret what's happening so that you can answer questions if given a picture like this. Okay? So what does it say to do? It says to start with fertilization, the fusion of two gametes, and trace what happens to the zygote. Okay? So here are our two gametes. So here are our haploid gametes. And if we look, we see we have two of these gametes coming together. And when these two gametes come together through fertilization, again, a gamete is a haploid egg or a sperm cell in humans. And we would call them just, gametes in just generalized organisms. So this one here specifically is a dinofat flagellate. So when these two form, we get a zygote, which is 2N. Okay. Now, this is very different from what we see in an animal life cycle because at this point now the zygote would go on to become a multicellular organism, and that would be it. But in this case, the zygote will undergo meiosis. Okay, it'll undergo meiosis, and when it undergoes meiosis, it'll go back to the haploid stage. These haploid cells will grow, get larger and larger and larger. Okay, they will undergo mitosis and make identical copies of themselves, and then ultimately join together to continue this cycle. So very, very different than what we've seen with animals, right? And even some plants in this alternation. Okay. Here's kind of a more, so this is where the haploid dominates. So the haploid is the one that grows. The haploid is the major life, the adult cycle, I guess you could say. Here we've got a cycle where the mature cell is diploid. Okay. So again, we want to start with our egg and sperm in this case, coming together through fertilization. Okay, here they are, fertilized together, egg and sperm. We got the full diploid number. Okay, so not much different so far. It's going to grow. We're going to get this initial cell. The mature cell now is going to be our dominant form. Okay, so this mature cell now will undergo mitosis. Okay, the 2N undergoes mitosis, whereas here the haploid underwent mitosis, excuse me. The mitosis will allow this mature cell to continue to reproduce and make exact copies of it, the 2N. In each division, both halves become the external template of the daughter cells. Therefore, one of the daughter cells will be smaller. So specifically in this diatom, we see that happening. All right. Undergoes mitosis, and we get exact copies. Okay. In this case, the smaller cells cannot divide anymore, and these are the ones that are going to undergo meiosis to produce the egg and the sperm cells. So again, very unique life cycle here. So you're not going to have to memorize this, but you should understand what's happening given this picture and be able to follow, like I said, from fertilization all the way through. So this type of life cycle where you're going back and forth between the dominant haploid form or the dominant diploid form back and forth is called alternation of generations. And you will see this again later on when we do plants. And you want to recognize this term gametophyte because the gametophyte is going to play an important role when we get to ferns. We're going to see it, and also some other plants as well. But alternation of generation, one phase of the life cycle is based on haploid, the other on diploid. Okay, so specifically the gametophyte is this multicellular haploid form. So really weird. You don't think about, a haploid cell like an egg or a sperm being multicellular, right? It's not something that we have. But in plants, we do see this multicellular gametophyte. And the specialized cells in the gametophyte produce gametes, single haploid cells, by mitosis and cell division. Okay, so the gametophyte is basically just going to be this giant sperm cell making more sperm cells. You can think of it like that. And then two gametes that are produced in the gametophyte will come together to make a diploid zygote. lot of times the gametophyte is, depending on what's the dominant stage, the haploid or the diploid, the gametophyte can be huge, or it'll be really, really tiny. The multicellular diploid form is the sporophyte. Okay, so this is the big 2N part of this alternation of generation. So the sporophyte would be the big fern. We're talking about ferns, and they do this life cycle. But the sporophyte contains specialized cells that produce spores, which are haploid cells, via meiosis. Okay, so haploid cells are then going to divide to form the multicellular gametophyte. So look at a picture of this here. Again, you want to understand what's happening. You don't have to memorize this life cycle. But if we, again, start with egg and sperm coming together, we get a zygote. Okay. Our zygote undergoes mitosis. We get more cells, more mitosis, and we get a sporophyte. Okay, the sporophyte is a diploid tree. Meiosis occurs in the sporophyte, and it produces these specialized structures here. Okay, these specialized structures undergo meiosis. Okay, and these specialized structures are ultimately going to be released as spores. And these spores are going to undergo mitosis. And when they do, they make a large multicellular gamete. Okay, and this gamete produces more egg and more sperm. So really an interesting thing, this alternation of generations. And again, we'll see this and study this in more detail in the coming weeks. We look at plants' life cycles. So very cool. So this is one where, notice the sporophyte and the gametophyte look exactly the same. But sometimes we'll see it where the multicellular haploid and diploid forms look very different. So here the sporophyte is large and the gametophyte is very small. Okay, but the same idea here, same life cycle, just different look to the gametophyte. It just depends on which is more dominant. So in this case, the sporophyte is the dominant stage, so the gametophytes will be the smaller ones. Okay, so let's look at some of the key lineages of protists and examine them in some more detail. So each of the 7 major lineages of all eukaryotes has at least one distinctive morphological characteristic, right? Things that kind of set them apart, which we've seen. But they've also greatly diversified in many different ways. Okay, there's been many different independent origins of parasitism, photosynthesis, multicellularity, right? And we see this really distinctive cell structure leading to that. Okay. And here's a list of some of the ones that we'll take a look at. So let's look at phylum Amoebozoa, or amoebas. So these Amoebozoa, these amoebas lack a cell wall. They feed by engulfing. They move by that amoeboid movement we talked about with those pseudopodia, excuse me. So what's included here in this phylum? We've got Lobos amoebae, cellular slime molds, plasmodial slime molds. Okay. So really kind of interesting. So if we look at these slime molds, right, they influence nutrient cycling by feeding on microorganisms. So really, these amoebas can be, very important in biological science. They have impacts as decomposers in forests as well. Okay, some are parasites of humans and other animals, which we talked about earlier in the last lecture. So, we have the acanth amoeba, brain-eating amoeba, all of these things that you find in freshwater habitats, wet soil. So some of these can be pretty nasty, but you want to, again, recognize the phylum Amoebozoa. We did talk about these in terms of their impacts on humans already. Kingdom Plantae, another example. Kingdom Plantae is a monophyletic group. So in terms of, looking at this group having one ancestor, Plantae have chloroplasts. They perform photosynthesis. The Plantae term is used to include the blue-green algae, red green algae, land plants. Okay. So specifically, the red algae are marine. Their cell walls have cellulose. They're photosynthetic, multicellular. Okay, these play a really important role in stabilizing coral reefs as well. Okay, so here's again some more information about red algae. I talked about this a little bit. Different types, 6,000 species. They're marine, obviously living in the ocean. Red pigment. This is the same. This phycoerythrin is going to produce the red color in red algae. Cell wall is made of cellulose, multicellular. Okay, asexual reproduction. The rhizaria, single-celled amoeba here. They lack cell walls. They move by amoeboid motion as well. And they produce long, slender pseudopodia, which you can see in this picture pretty easily. Foraminifera, we talked about them having that calcium outside of them. Actinopods are a part of this group as well. Okay. Asexual reproduction is common. Sometimes sexual reproduction, depending on if the conditions are allow for it. Okay. Alveolata. So these guys, the alveolata, are characterized by having membrane-bound vesicles or sacs under their plasma membrane. So the alveoli in your lungs are these sacs, and that's where the name comes from. They're unicellular, diverse in morphology. Some are capable of bioluminescence, so you get that beautiful bioluminescent sea, the blue-green sort of lit up ocean. You see sometimes pictures really pretty from like California and stuff like that, where you see that bioluminescent activity. Emits light from an enzyme catalyzed reaction. Luciferin, luciferase is the enzyme and the protein involved in that. Okay. Examples of organisms that fall under this phylum are the dinoflagellates. Dinoflagellates we talked about already. And here's the information about the dinoflagellates. So these heteroconta Stramenopila organisms here, these protists, these kind of are unique because they have this like hairy flagellum at some point in their life cycle. They include water molds, diatoms, brown algae. Okay. And you can see some of the traits here. The diatoms we saw earlier, they have that glassy cell wall. They're planktonic, photosynthetic, unicellular. Okay, brown algae is multicellular, composed of leaf-like blades, sessile and photosynthetic. Brown algae. Take a look at pictures there of those and the information in these tables. So you do want to, go through and review some of the stuff in the chapter, any supplemental videos that I had for chapter 27, and I will see you next time. Thank you for listening.